friends, in today's session we'll start with the second chapter, that is sexual reproduction in flowering plants. So before that, if you didn't subscribe our channel, please do subscribe and press the bell icon for further updates. So we'll move on to the session. In today's session we'll talk only about the male reproductive part of the flower. So this is the male reproductive part of the flower. So now when you take the LS of a flower, what do you call it as the male reproductive part? The anther and the filament. So filament is just the tubular structure and onto which anther is then attached. Now any structure which consists of anther and filament, you call it as stamen. So basically the male reproductive part is just the stamen in the flower. And now when you want to indicate a male flower, just don't indicate as it has anther or filament, just you can say it as it is a staminate flower. So what do I mean by staminate flower is that it is a male reproductive part of the flower, or it is the male flower, the flower consisting of the male organ, and you call it as a staminate flower. So this is the first point. Next, filament is just the tubular structure. There is not much detailed explanation on that, but when you talk about anther, there are so much to study about anther. Now the first point is that anther is a bilobe structure. Bilobe, which means it has two lobes. Now this is the first lobe and this is the second lobe. 60% of angiosperms, that is the flowering plants, they have bilobed anther. Alright. Next you have each lobe has two thecas. Now each of these lobes will have two separate compartments. Compartment number 1 and 2, and here in the second lobe compartment number 1 and 2, and these compartments you call it as thecas. So each lobe has two thecas and you call it as dithecous. Now one lobe has two thecas. Now imagine a filament has bilobed anther. So totally we have four thecas. Alright. Next, it is a tetragonal structure, which means that it has four corners. Four corners, and you call it as a tetragonal structure. Now you have longitudinal grooves. Grooves are nothing but a line or a cellular structure that differentiate each of these lobes, right? And now what happens is that now you call it as four microsporangia. So each of the lobes, so four thecas you have, each of these thecas will have microsporangia into which the pollen grains develop. So this is the structure of anther. Now pollen grains develop inside the pollen sac. Now this pollen sac is present in the microsporangia. Now we'll see where is this microsporangia present. Alright. So that is why we are studying the structure of microsporangium. Right. Now this is just a slit structure of a transverse section of a pollen grain. So when you take a TS of a pollen grain, this is how it looks like. So it is a microscopic structure. This, like, to indicate only the cell walls have drawn like that, but when you see it in your book, you have a lot of cells in them. So that's how they exactly look under the microscope. So first they have four walls: epidermis, endothecium, middle layer and tapetum. So the first layer is epidermis. Now the second layer is endothecium. The third layer is the middle layer, and the fourth layer is a spherical layer called as datum. Now first, all these three layers are just for the protection, to protect the pollen grains from environmental disaster, right? And now the fourth layer, tapetum, they provide energy and nourishment for the pollen grains. They are the precursors which they store the energy which is required for the pollen grains to develop by the process of microsporogenesis. So that's it for today's session. We saw what is the male reproductive part of a flower, the structure of anther, and the structure of microsporangium. So in next class we will talk about how the pollen grains are formed by the process of microsporogenesis. If you want to take a screenshot, you can take it. Thank you. In class we will talk about Microsporogenesis, which is the production of pollen grains and the formation of pollen grains. So before that, if you didn't subscribe our channel, please do subscribe and press the bell icon for further updates. So we'll move on to the session. Yes, microsporogenesis, micro. When you talk micro, it is a male reproductive part. When you say it as mega, it is a female reproductive part. Alright, so it is a male gametophyte. Now what is microsporogenesis? It is the formation of pollen grains from PMC, that is pollen mother cell. Alright, and now we'll see the structure of pollen grain. So the structure of pollen grain, they vary from plant to plant. Alright, some could be spherical, elongated structure, and some could have a spike. So when you take a proper examination of scanned electron microscopic picture in the net, you would be able to see how wonder the nature. Alright, so now we'll talk about the different layers of the pollen grains. Now pollen grains have two layers. One is exine and the other layer is intine. Now exine is the outermost layer and it is made up of sporopollen. So sporopollen is an organic material which is covalently bond. So basically the pollen grain is made up of this harder coat. It's to make it withstand in a harder environment. For example, if the wind blows really forcefully, the pollen grains should not shed off that easily. So for that purpose, this sporopollen, the exine is made up of sporopollen. And the next second structure, the inner structure, is made up of cellulose and pectin, and this layer is called as intine. Alright, exine the outer, intine the inner. Now what happens, you have the nucleus and the vacuoles in the cytoplasm. So vacuoles are generally present to store energy, to provide energy for this microsporogenesis process. And now what happens, this pollen mother cell, they divide into two cells. One is the vegetative cell which floats on the surface of the cytoplasm. Next is the generative cell which settle towards the poles of any one of the poles of the pollen grains, right? So look, it could be either here or it could be either here, right? So poles. So but vegetative cell can freely float on the surface of the cytoplasm, generative cell. Now you have vegetative cell and generative cell. So 60% of angiosperms are always in a two-cell stage before it has been shed off, right? But now what happens, in other 40% of the angiosperms, the generative cells again undergo mitotic cell division and form two generative cells. So there is a cell, see there is vegetative cell. So one vegetative cell. Now this generative cell will undergo mitotic division and form two generative cells. So you have cell 1, cell 2 and cell 3. So that is, it forms 3 cell stage in other 40% of the angiosperms. So either in a 2 cell stage or the 3 cell stage, the pollen grain is now ever ready to shed off and fuse with the ovary, right? And now what happens, and the viability. So viability is the potentiality of it to, or the vigor of it to fuse it with the ovary. So that period you call it as the viable period, right? And now this viability could range from 30 minutes, for example rose plants, or nearly months in wheat plants. So anything between 30 minutes and months, nearly months, 2 to 3 months. So it could be 1, 2 and maximum of 3 months. It is the average range for it to keep up its own viability. So now we saw about what is microsporogenesis, structure of pollen grains. So now pollen grains is much high in nutrition. So what happens, they collect these pollen grains and sell it in the form of bee pollen or pollen liquids in a syrup form, and which is much helpful for you to increase your metabolism and give you a lot of energy. So athletes will use all this type of pollen grains, pollen syrups. And now what happens, if you think this pollen grain is highly viable in nature, it has the potentiality, so people, they collect these pollen grains and store it in liquid nitrogen in the temperature of minus 196 degrees Celsius. So the major function of a liquid nitrogen is to arrest the cell division. So now what happens, either if it is in the 2-cell stage or 3-cell stage, it arrests the cell division and maintain the pollen grains and the nature of the pollen grains as such. Alright, so that is how you store, and later after 1 or 2 months you take it out, you maintain it in the room temperature and make it fuse with the ovary for the production of fruit, flowers and fruits. Alright, so in today's class we saw all this, and in tomorrow's session we'll talk about the female reproductive part of the flower and megasporogenesis. Thank you. If you want to take a screenshot, you can screenshot it. Today's class will talk about the structure of ovule. So before that, if you didn't subscribe our channel, please do subscribe and press the bell icon for further updates. So now come on, we'll move on to the session. Yes, now what exactly you call it as the female reproductive part of the flower? It's all because of 3 parts. One is ovary, style and stigma. So ovary is always a spherical or a globular shaped structure onto which attached a style, which is a tubular structure, and onto which you have stigma. So the major function of stigma is to hold on the anther properly. They are adhesive in nature to hold on the anther and grab it inside the style and then eventually to the ovary. Now when there is a female flower, you call it as a pistillate flower, and all these 3 structure you group it under pistil. So a female flower, you call it as a pistillate flower. Now this ovary has ovules, so there could be 1 ovule or many, more than 1 ovules. And now we are going to see 4 important terms related to NEET exams, and we'll see one by one. Now first, when this pistil is so isolated, it has a ovary, style and stigma, you call it as monocarpillary. You indicate this condition by the help of this term, monocarpillary. Next, when you have more than 1 pistil, so when there are like so many structures this way attached together, you call it as multicarpillary. Now when the pistil is free, so this is a free pistil, right? It is not disturbed by any other pistil, so that you call it as syncarpous. Apocarpous is fused pistil, where there are many pistil fused together, and this term is indicated by apocarpous. So this is an introduction about ovary. Now we'll see a detailed structure of the ovule. Now what happens, this ovary has a cavity, and this cavity is called as ovarian cavity. Now inside the ovarian cavity you have something called as placenta, onto which the ovule is being attached. Clear? Now the ovule is attached to the placenta by the help of funicle, but how exactly it is attached is because of helium. So helium is a stronger structure that supports funicle to hold on to the placenta. Alright, now when you talk about the structure of an ovule, it has two layers. One is the outer layer, the other is the inner layer. The outer layer you call it as the outer integument, this part, and the inner layer is inner integument, this part. Alright, now these two parts are attached to a group of cells called as nucellar cells. Nucellar cells, and you call it as nucellus. Now inside the outer integument, inner integument and nucellus, there lies an embryo sac. Now this is exactly the female gametophyte, right? And now what happens, this whole structure of an ovule has two poles. It has two poles. Now the bottom poles, which is right opposite to the funicle, is the chelacal pole, and which is adjacent to the funicle is the micropyle, and you call it as a micropylar pole. Now it has outer wall, inner wall supported by nucellus, inside which you have the embryo sac, which is exactly the female reproductive part of a flower, and then you have two poles, the micropylar pole and the chelacal pole. So in next class we'll talk about megasporogenesis, how exactly the ovules are produced. Now if you want to take a screenshot, you can screenshot. Class we spoke about the structure of ovule. So before that, if you didn't subscribe our channel, please do subscribe and press the bell icon for further updates. So yes, so now first we'll talk about megasporogenesis, which means the formation of the megaspores from the megaspore mother cell is called megasporogenesis. Now first, what exactly is that these megaspore mother cells, they form 4 cells, and out of which 3 cells will disintegrate and only 1 cell will move further, and now this is called as monosporic development. Now what happens out of these 4 cells, when 2 cells disintegrates and 2 cells are functional, you call it as bisporic development. The same way you have trisporic development and tetrasporic development. Alright. Now what exactly happens in the nucleus of megaspore mother cell, we are going to talk today. Now first, the megaspore mother cell has a single nucleus. Alright. Now this nucleus will undergo mitotic division and lead to 2 other nuclei. And now again this so it is not cytogamy, only the karyogamy, only the nucleus they divide. So that you call it as karyogamy. Alright. Now again this will undergo mitotic division, and now it is 4 nuclear stage, and again each of this, so now you have this 8 nuclear stage. Now how exactly this 8 nuclear stage, they assemble themselves inside the embryo sac is what we are going to talk today, right? Now what happens, this ovule has two poles. One is the chelacal pole and the other is a micropylar pole. Now in chelacal pole, 3 nucleus goes and attaches. In micropylar pole, the other 3 nucleus goes and attaches. And in the middle of the cell, you have polar nuclei. Now all the nuclei, they are surrounded by cell wall except the polar nuclei. So now we have 8 nucleus. 1, 2, 3, 4, 5, 6, 7 and 8. So you have 8 nuclear stage, but it is called as 7-celled stage. Why? Because any cell should have its own proper cytoplasm surrounded by the cell wall. Now 1, this has its own cell wall. 2, 3, 4, 5 and 6. But what happens to the polar nuclei is considered as a single cell because they are not surrounded by cell wall. You consider this region as its cell wall, right? So it has 8 nuclear stage with 7-celled stage. Okay. Now what exactly happens? Now in the chelacal pole, you have 3 nucleus and you call them as the antipodal nucleus. So 3 nucleus. And now in the micropylar end, you have other 3 nucleus. The first two nucleus is present between the synergids, which you call it as they are all the energy producers of the cell. And now you have this filiform apparatus. This filiform apparatus helps the anther drive through the pollen tube and fuse it with the ovary. So for this purpose, this filiform apparatus is useful. So now next is the egg cell. Next you have polar nuclei, which is present in the middle of the cell, central of the cell. Alright. So now 8 nuclear stage with 7 cells. Right. So this is all about the assemblage of the nucleus inside the embryo cell. So in next class we'll talk about pollination and how this pollen grains, they come in contact with the ovules and fuse, and what are the steps that will happen further. Thank you. If you want to take a screenshot, you can screenshot it. Move on to the session. Yes. Now pollination. So now what exactly is pollination? The transfer of pollen grains from the anther to stigma is called as pollination. In other terms, there has to be an interaction of the gamete from one flower to the other flower. Alright. So a male flower, you call it as a staminate flower. A female flower, you call it as a pistillate flower. So now what exactly happens? The pollen grain from anther of a flower will go and interact with the stigma of the female flower is called as pollination. Clear? Now next we'll talk about the kinds of pollination. Now what are the different kinds of pollination? The first thing is self-pollination. So the term self is within the flower. You call it as autogamy, which means when the pollination happens in the same flower, in the same single flower, you call it as autogamy. For example, this single flower will have stamen as well as a pistil. Right? Now the transfer of pollen grains from anther of a flower to the stigma of the same flower, that you call it as autogamy. Next is cross-pollination. Cross-pollination of two different types. One is happening in the same plant and the other is between different plants of same species. Now same plant cross-pollination, you call it as gatogamy. Now what exactly is gatogamy is that, now imagine a single plant, a rose plant. Now any plant you can imagine. Now this is a male flower and this is a female flower. So this is a staminate and this is a pistillate. Now what exactly happens is that the anther of this male flower and this is the pistil, the stigma of the female flower. Now the pollen grains from the male flower is transferred to the female flower of the same plant and you call it as gatogamy. Now next type of cross-pollination is xenogamy, which means between this different flower, different plants of the same species. Imagine there's a two flowering plant, right? Now this plant, you take this flower and this plant, we'll imagine this flower. Now this is a male flower and this is a female flower. Now what exactly happens? Now the transfer of this pollen grain from the anther of this flower to the stigma of another flower of another plant is called as xenogamy. So that's the concept of different kinds of pollination. So next we'll move on to two different types of flowers. One is chasmogamous flower and the other is clistogamous flower. So chasmogamous flower is a normal flower which has both male and the female reproductive part, where there is a transfer of anther from the male part to the female part happens. But in case of clistogamous flower, imagine this is a bud. right? The flower hasn't bloomed. This is a bud. Now what exactly happens? But in case of casmogamous flower, it is a bloom flower. So any type of pollination is possible in casmogamous flower. But in clistogamous flower, which do not open, there is a possibility of autogamy alone, which is the anther, the stamen and the pistil. Interaction will happen only in the flower, inside the flower, because there's no chance of cross pollination. So that type of flower you call it as clistogamous, and the other type of flower you call it as casmogamous flower. Next we'll talk about the agents of pollination. What are the agents that helps in the transfer of pollen grains to the female flower, right? The first is abiotic agents, is the non-living agents, and biotic agents, of course the living agents. Now non-biotic, like abiotic agents, you have wind and water. So when it happens because of wind, you call it as anemophily, and water hydrophily, right? When the pollination occurs with the help of wind. Now imagine there's a force wind that has been forced up or blowing up on the flower or on the plant. Now what exactly happens? Because of the interaction of this wind, what happens? The pollen grains of the anther, they tend to wither down and spread it to the stigma of the female flower. So that exactly in the case of all the types of phyllies, right? Now water, you have hydrophily. So these are the two important abiotic agents. Now in biotic agents, so any pollination that happens because of animals, you call it as zoophily, right? Now in that specifically, when it happens because of snails, you call it as melacophily. But now all these terms, this one and this kinds of pollination, you can expect in your competitive exams and your NEET exams, right? So you better you like study all these terms. So because of snail, you call it as melacophily, and when it happens because of insect, you call it as entomophily, and when it happens because of birds, it is ornithophily, and in the last batch, you call it as chiropterophily, right? So in today's class, we spoke about what is pollination, the kinds of pollination and the agents of pollination. In the next session, we'll talk about the interaction between the male and the female gamete. So if you want to take a screenshot, you can do it. Hello friends, so last class we saw about the pollination and the agents of pollination, and today's class we are going to talk about what happens after pollination. So that is the pollen pistil interaction and the fertilization process. So first we're going to talk about pollen pistil interaction. So last class we spoke about pollination, right? So if you're clear with the concept, this is very easy to understand. So now what exactly happens is that pollen tube. Now the pollen goes through the pollen tube and meet the ovules. Now how does this happen? Now the pollen tube is infused in the tissues of style and stigma, so that they create a pathway to reach the ovule. So this is the basic process. Now there is something called as compatibility and incompatibility. So now when the pollen grains of the same species fuse with the ovules of the same species, it is said to be compatible, right? So the pistil accept the pollen grains, and this is called as compatibility. In few cases, in terms of pollination, there are chances of the cross mixing of the pollen grain to the ovules of different species, and this concept is called as non-compatibility. Now in this case, the pistil rejects the pollen grains. So just remember, only if it is of a same species pollen grains, the pistil accepts. If it is of different species, the pistil rejects, right? And next, now what happens is that now there is a pollen grain. Now it is traveling through the pollen tube. Now this pollen tube is infused in the tissues of style and stigma, which is the female reproductive part. Now alright, now what happens? The synergids. Now we spoke about the LS of ovule, right? I'll draw it even more clearly. Now you have ovule. Ovule has two poles. One is the chalacil pole and the micropylar pole. The entry happens only through the micropylar pole. Now you have three antipodals, two polar nuclei, one egg, and two synergids with filiform apparatus. Clear? Now what happens is that the pollen grain, so I told you, right, 60% of the pollen grains is shed off in two nuclear stage. One is the generative nucleus and the vegetative nucleus. Now these two is considered as two gametes, two male gametes, and now there is a single female gamete which is in 8 nuclear stage, and now the pollen grain is in 2 nuclear stage. It's almost like 10 nucleus. Understand? Now what happens? Now after this process, the germination starts to happen. After pollination, the germination starts. But between this pollination and germination, there's a very small important process called as fertilization. And in case of this reproduction in flowering plants, there's something called as double fertilization, because the fertilization happens twice, 2 times. Right? Now what happens? I told you, the synergids, the filiform apparatus will guide the pollen grains through the pollen tube to the ovules, right? And now that's the function of filiform apparatus. Now synergids will provide the energy for the fertilization to happen. Now two gametes, right? Now one of the gamete, that is the generative nuclei of the pollen grain, will go and fuse with the egg. So now the generative nuclei is haploid, egg is haploid, so you get a diploid zygote. Clear? Now there is another vegetative nuclei of the pollen grains. What happens? The vegetative, this is in case of generative nuclei of the pollen grains. Now the vegetative nuclei of the pollen grains will go and fuse it with the polar nuclei. Now already polar nuclei has 2 nucleus. Now there is one more vegetative nuclei, which is 1. So you have 3 nucleus altogether, and now this you call it as a triploid pen. Pen means primary endosperm nucleus, right? Now first, the generative nucleus goes and fuse with the egg, and the vegetative nucleus go and fuse with the polar nuclei. Now the fertilization happens twice, and this process you call it as double fertilization or dual fertilization. So first is the fusion of egg, next is the fusion with the polar nuclei, and now the pollen grain, which is the nucleus of the pollen grain which is fused with the egg, will turn out to be the zygote, which is the embryo, and the other term, the polar nuclei, this will convert into endosperm. So next class we'll talk about endosperm, zygote and seed. If you want to take a screenshot, you're welcome. Friends, so last class we spoke about pollen pistil interaction and what happens during the process of double fertilization. In today's class, we are going to talk about endosperm and embryo formation. After fertilization, how this embryo and the zygote is been formed. So before we move on to the session, so yes, first we'll talk about endosperm and then we'll talk about embryo. Right? Now what happens is that last class I told you there's a pollen grain. It has a vegetative nucleus and a generative nucleus. Clear? And now what happens? This vegetative nucleus, they go and fuse with the polar nuclei. Clear? Now this polar nuclei along with this vegetative nuclei is what is 2-pen stage. Correct? Now what happens after fusion? Now all the 3 nucleus has been fused together, is compiled together. After this stage, what happens is there is a repeated nuclear nuclear division, which is called as the karyogamy, right? Now what happens after repeated n number of nuclear division, each of this nuclei tends to be surrounded by cytoplasm. So this is what happens after the fertilization in related with the vegetative nuclei of the pollen grain. Now the peep seed, the groundnut and the beans which we cook and eat are nothing but the endosperm of the plant. So it is not that hard in nature, but before the formation of embryo there is always the formation of endosperm, because this endosperm stores the nutrition for the development of embryo. Now you are in this tender coconut, there is a coconut water, right? Now what exactly is this coconut water? There are thousands and thousands of karyogamy that has happened after this two-pen stage. So that is what called as this coconut water, and it is very good for health, right? And next we'll talk about embryo, the formation of embryo. Now what exactly happens? I told you about the vegetative part. Now this generative part will go and fuse it with the ovule, right? And now what happens is everything happens in the micropylar end. So at the end of the micropylar end, there is a formation of a precursoral component of the embryo called as proembryo. After this proembryo, there is a formation of zygote or the embryo. Now we'll talk about the embryo of two different plants, the monocotyledon plant and the dicotyledonal plants. So in monocots, you have in dicots, you have two cotyledons, which means that you can split the cotyledons into two equal halves. So that is two cotyledons. Now in such cases, the important part of an embryo is the embryonal axis. The region above the embryonal axis is the epicotyle, and the region below the embryonal axis is the hypocotyle. Above, epi, below and hypocotyle. Now this epicotyle will start developing into the shoot system, leaf, flower, stem and everything above the soil, that is your shoot system, and everything below the soil is your root system. So that is developed from the hypocotyle, where the root along with the root cap, right? And in case of monocots, where you can never split the cotyledons, it is just a single cotyledon, and this term is very important for your competitive exam, and we call it as scutellum, right? Scutellum. And now what happens in such cases of monocots, the embryonal axis, the region above the embryonal axis is the coleoptile, and below is called as coleorhiza. This coleorhiza, there is a root, formation of root, but coleoptile, why we are naming it coleoptile instead of epicotyle is that epicotyle shoot system, they develop into the different parts of the plant or a tree. But in case of this coleoptile, when you take grass plant, there is just a elongated leaf-like structure, and you call it as a coleoptile, right? So this is all about your coleoptile and the coleorhiza, the root system. So in tomorrow's class, we'll talk about the formation of seed. If you want to take a screenshot, you're welcome. Friends, so last class we spoke about the formation of embryo, and in today's class we'll talk about how exactly the seed is formed. So before that, if you didn't subscribe our channel, please do subscribe and press the bell icon for further updates. So we'll move on to the session. Yes, now seed. How seed is formed. So before that, we'll see the structure of a seed. A seed has three structures. The first is a seed has a coat, that is called as a seed coat. Next, it has cotyledon. It could be a single cotyledon or a bicotyledon, where a cotyledon which cannot be split into two equal halves, or a cotyledon which can be split into two equal halves. Next, you have the embryonal axis from which the epicotyle and the hypocotyle developed. Alright, so this is the structure of seed. And now seed, you have two important types of seed. One is non-albuminous seed and the other is albuminous seed. Non-albuminous seed do not consist of a residual endosperm. So last class I told you all, example pea plant and groundnuts, the thing which you cook and eat is the exact endosperm. So there is no residual endosperm. It is the endosperm, right? So that is called as non-albuminous seed. Now in albuminous seed, you have endosperm at the micropilar end above which the seed is developed. So this is called albuminous, and for example wheat, maize and barley plants, right? And now we'll talk about what is a perisperm and a pericarp. Now on the ovules you have the nucellar cells, right, above which you have the inner integument and the outer integument. So if you would have seen my previous session, you would have known the concept. So nucellar cells, when it starts dividing, so the nucellar cells will become the perisperm of a fruit. Next is how a seed is formed and a fruit is formed. Now the ovules form seed and the ovary will form the fruit, and this fruit is called as the pericarp. And again this pericarp you can divide it into endocarp, mesocarp and the exocarp. Now imagine this is the section of apple fruit. Now inside you have the seed, right? The space just above the seed is the endocarp, and next this fleshy part is the mesocarp, and the outer coat is the exocarp, right? And now very simple you can remember is that there is ovary, you have ovules. Ovules develop into seed and ovary develop into the fleshy part, fruit. And now we'll talk about parthenocarpic fruits. And now when a fruit is formed without the process of fertilization, you call it as parthenocarpic fruits. Example banana plant, and this can be induced by applying growth hormones on them. So banana plants, they basically reproduce by vegetative propagation. So previously I spoke when I was talking about vegetative propagation, I told you a process called as a sucker plants, right? In that I gave you banana as example. That is one such reason for the formation of parthenocarpic fruits. And next we'll talk about apomixis. So apomixis is the seed without the fertilization. So usually the gametes are haploid. The ovule is haploid and the pollen is haploid. But in terms of apomixis, the ovary will not undergo reduction division. Rather, the ovary is 2n in number. It is diploid in number, and this induces the ovary to form the seed. So that process is called as apomixis, the mixing of two haploid ovules. Next, we'll talk about polyembryony, which means many embryos. How many embryos are formed? When the nucellar cells continuously they divide, the ovules they become embryos. There are many ovules right inside the ovary, and each of these ovules will become a distinct embryo. So this could be seen in case of citrus plants like oranges and mango. So that is called as polyembryony. See, parthenocarpic fruits, no fertilization. Apomixis, seeds without fertilization, that is the diploid ovules. Next, polyembryony, continuous division of the nucellar cells, and the ovules will develop into n number of embryos. So that's all, we are done with the chapter, and tomorrow we'll start with a new chapter. So if you want to take a screenshot, you're most welcome.